Okay. Well, welcome to the course. I apologize again for having taken too long to figure out how to start and how to put things for you on the system. But hopefully today we can start. I also thought that if the students weren't present in the class, I shouldn't give the class. But now I was told that the recordings are enough, even if a small number of students manages to connect. So welcome, Farah. I see you are there, one of you. And now we need to catch up a little bit because we missed 4, I think, or 5 sessions for a total of at least 12 hours. And so we're going to make up in the evenings. I am thinking of, on Monday when the classes are not scheduled, we'll have a class Monday from 6 to 8. Tuesday as well will have to be from 6 to 8 because now I have seminars at the university until 5 in the afternoon. So Tuesday is going to be moved to 6 to 8. Wednesday also we do 6 to 8, and then Thursday we do the 4 to 7, and Sunday we do 4 to 7. So like this, hopefully we will manage to catch up with the material. So I will give a class tomorrow at 6 o'clock in the evening. Now, I put on the ITC the PowerPoint presentation and the outline for the course. So let me know if you have any difficulties with opening or downloading or getting hold of that material. And the other thing is this course always has also a laboratory component. Now, we can't do laboratory work, obviously, but I will do, at the end, I will do one Sunday where we spend all the time looking at laboratory work. I might do some practical and recording them so that I can send them to you for how you carry out some microbiology lab activities so that you will have a kind of a virtual access to the laboratory. Okay, so let me start. Now I live in Birzeit. I have been living in Palestine for the past 20 years. I am originally Italian. I married a Palestinian and we moved here 20 years ago. I speak a bit of Arabic. I mean, I speak colloquial Arabic enough to understand and be understood, but my Arabic is not at the level of being able to give a scientific class in Arabic. So this course is going to be in English. But if you need to you need for me to repeat anything, or if you don't understand anything, or if you have any questions, please feel free to put your hand up and I will see it, on the Zoom, there is this hand up facility. And so I can explain things again and we can discuss. clarify what might be not clear for you. So if you don't have any questions, I think we should start with the lecture, and we start with the bacterial cell structure. So we dive right into the bacteria and what is their structure and what are the general characteristics of bacterial cells. Now we know bacteria are prokaryotic cells. There is a message. You can't hear me? There is no sound? Why? The sound is on. Can you hear me? Can anybody let me know if you can hear my voice? Okay, I'll carry on, and then if there are any problems, you will have the recording. So the bacterial cell structure, bacteria are no. Okay. Can you hear me now? Yeah, you can hear me? Okay. Right. So I just summarize what I said before. Again, I am Italian in origin. I moved to Palestine 20 years ago. My Arabic is not at the level for me to teach in Arabic, but if you have any questions and if you want to ask anything, even if you ask in Arabic, I should be able to understand you. And if you don't understand anything, please stop me at any point and ask. Bacteria. Bacteria are prokaryotic cells. What does it mean that they are prokaryotic cells? Prokaryotic cells are cells that have a fairly limited organization in terms of architecture of the cell. They are described usually as cells that don't have a nucleus, meaning that the DNA, the chromosome of bacteria is not surrounded by a nuclear membrane, but is free in the cytoplasm. It is not only the nucleus, in fact, that is missing in bacterial cells, but we can say also that prokaryotic cells in general don't have a internal system of membranes. So those structures like the endoplasmic reticulum, for instance, the Golgi apparatus, lysosomes, vesicles in general are not found in bacteria. And we don't also have organelles like mitochondria or chloroplasts. So the bacterial cell is a cell that is in architecture, in the way that is built, quite simple. It doesn't mean that prokaryotic cells don't have the ability to carry out complex and sophisticated metabolic functions. It's just that they do it in a way that is different from the way that is used by the eukaryotic cell. Actually, not having these internal organelles and not having internal membrane compartmentalization inside the cell gives them a little bit more flexibility, which actually allows them to have more than one way to carry out specific metabolic activities. And we will see later in the course how these make them quite efficient actually at carrying out metabolism. And so let's start with the bacterial cell structure. And we start by looking at some general characteristics of shape, arrangement, and size. So the shape, the form they have, the arrangement, how they can form specific groups of cells, and what is their size. Now, shapes. Bacteria are a very large group of organisms. In fact, if you remember probably from other biology courses that you have taken, bacteria make up a whole domain of classification. You know how all cell life is organized into 3 domains. You have the bacteria, you have the archaea, and you have the eukarya. So bacteria are a large group of organisms. So far, the bacteria that have been isolated and identified have been organized into 40 phyla. And you know how each phylum then has order, class, family, genus, species. And so the number of species of bacteria is very high. We have so far isolated a very large number of bacteria. And so you can imagine that having this large number of organisms, we find very many different shapes, which are present in nature. Some shapes seem to be more common than others, and the coccus shape and the bacillus, coccus is round and the bacillus is rod shape, are the most common shapes. Now, somebody has told me again that you cannot hear me, but before you said that you could hear me. Sorry, some people we can't hear you. Maria. Nobody can hear? Oh, this is strange. Can you hear now? So Farah can hear. So maybe the problem is not on my end. Farah can hear, so I think that maybe other people have, maybe they need to do something with their... Okay, so coccus and rod are the most common. Coccus is the round shape and rod is a bacillus shape that is like a little stick. Yeah, but there are others. The arrangement depends on how the cells behave after they divide. When there is cell division, cells divide. If they separate and each cell goes on its own, there is no arrangement. But sometimes they don't separate after cell division and they stay connected. And this connection can form different arrangements. And we're going to look at them in later. The size, of course, again, having so many different species, you have also many different sizes. Is generally small, you know, they're microorganisms. Microorganisms are small by definition. In order to observe microorganisms, you need to have a microscope. And so you have very many sizes, but all of them very small in the micrometer range. Yeah. Now, here we have the coccus shape and arrangement. Coccus is the singular, cocci or cocci is the plural. They're spheres. They're really, really very nice, almost perfect spheres. Yeah, you can see them here. These pictures are taken with a scanning electron microscope, and you can see very well the pearl-shaped coccus. Now, when cocci divide, they can stay attached to each other. And if it is only two cells that are attached to each other, you call this diplococcus. Di means two, yeah, it's paired, diplococci or diplococcus. Strepto is the prefix that is used to indicate chains of bacteria, Sincella, yeah. So Streptococcus is like here, you see in this figure, in this picture here on top left. They are organized in a chain, Streptococcus. And then you have the Staphylococcus. Staphylococcus, the cells they attach to form clusters of cells that are similar to grape. Yeah, Zellane. If four cells stay together, we call that a tetrad. And when you have four cells together with four on top, they make like a cube, yeah? And that is called a sarcina. But again, Streptococcus and Staphylococcus is the most common. Diplococcus can also be quite common, especially with the lactobacillus. Yeah? But as I said, even though these two shapes are the most common ones, there are other shapes that are possible. You have, oh, here, sorry, we didn't talk about the rod. Here is the rod, yeah, the bacillus shape. We have a vibrio. Vibrio is a comma shape. Here, vibrios. You have Coccobacillus. Coccobacillus are bacilli, so they are rods, but they're so small that they look as if they were round, but they're not round, they're rods. So they're very short rods. Then you have the spirilla, and you have the spirochete, yeah? Spirilla, like this one here, which has it's like a helix. They halazone, but is quite rigid, or you have the spirochetes, which are also halazone, but they are more flexible. And then there is stalk, there is mycelium, there is also bacteria that don't have a fixed shape. Bacteria that don't have a fixed shape are called pleomorphic. Now, there isn't many of them that are like this. Why? Because most bacteria, 99.9% of bacteria, have a cell wall, and the cell walls make the cell rigid. Yeah, the wall is a rigid structure, and so the shape becomes fixed if you have a cell wall. Okay, but some of them don't have a cell wall. These are the ones that for instance, live inside other cells. So they're protected by being inside the cytoplasm of another cell, so they don't need a cell wall. And because they don't have a cell wall, like animal cells, their shape is not fixed. Size, as I said before, size changes. They are all very small. So you need a microscope to observe them, but you have some that are very, very small, like mycoplasma, and mycoplasma is an example of a bacterium that is parasitic and lives inside the cytoplasm of other cells. The average rod is 1.1 to 1.5 micrometers, 2 to 6 micrometers. Yeah, the width is 1.5 and the length is 2 to 6. Yeah. There are some that are very large, but they are not very common, like this Epulopicium fischelsoni, sorry, which is 600 micrometer, almost a millimeter long, but they are exceptions. Both very small and very large are exception. The average size is 1 by 2 or 1.5 by 6. Yeah. So here is E. coli is 1.3 by 4. 1.3 is the width and 4 is the length.
Hi, Miss.
Yes. Hello.
This is the first course.
This is the first class.
Yes, I am not late.
You're not late. No, I was late. I apologize. And we will make up. As I said, we will have a class tomorrow as well from 6 to 8. I'm going to put a schedule on the course board. Now we have a class schedule on Tuesdays from 4 until 7, but I have seminars now at Birzeit University until 5. So Tuesday is also going to be from 6 until 8. So I will put a schedule on the ITC site, and hopefully we'll manage to catch up and finish on everything on time. Okay, so now that we understand roughly the idea of the shapes and the size, we move into looking at the cell organization. As I said before, prokaryotic cell and bacterial cells are quite simple in their structure. They don't have internal membranes. They don't have internal organelles. There is no endoplasmic reticulum. There is no nucleus. There is no lysosome. There is no Golgi apparatus. There are no mitochondria, no chloroplast, nothing of those structures that can be found inside the eukaryotic cell. So a much simpler organization. If we look at this drawing, we have a schematic representation of what is found in a bacterial cell. You have the chromosome. It's one chromosome, yeah. which is free in the cytoplasm. This is the genetic information. You have ribosomes, of course. Ribosomes are essential for making proteins. So every cell must have a cell membrane, DNA, and ribosome as a minimum to be a cell. Yeah. And then you have inclusions that might be there or might not be there. You have the cell membrane, you have the cell wall, and then you might have other structures outside, like a capsule. You might have the flagellum, you might have fimbriae. These are not always there. What is always there, that is an essential requirement to have a bacterium, is the cytoplasm, ribosomes, the DNA, the cell membrane, and the cell wall, because the great majority of bacteria have cell wall. This is all that there is. So 5 things, yeah, much simpler compared to a eukaryotic cell. But as I said earlier, this simplicity doesn't mean that the cells are not good at metabolic activities. Actually, they can be better in some cases compared to eukaryotic cells, because not having those organelles give them more flexibility in their metabolic activity. So we're going to start by looking at the cell envelope. So what is that contains the cytoplasm? Yeah. We have the plasma membrane, we have the cell wall, and as I said before, plasma membrane and cell wall. Plasma membrane, 100% of cells must have a plasma membrane. If you don't have a plasma membrane, you're not a cell, okay? So the plasma membrane is always there. The cell wall, when it comes to bacteria, as I said, 99.99% of bacteria have a cell wall, but there is a very small number with no cell wall, and these are the ones that are parasitic, the ones that live inside other cells. But free bacteria must have a cell wall. And then there are layers outside the cell wall. This layer outside the cell wall may be there or not. They are not a requirement, but several species have it, which is the capsule, the slime layer, and the S layers. We're going to talk about. So let's start by looking at the plasma membrane and what is the plasma membrane function. Again, a plasma membrane is, by definition, the structure that surrounds the cytoplasm. It goes around the cytoplasm and holds the cytoplasm inside the cell. Yeah, so it is the structure that contains the cytoplasm. As I said, it's an absolute requirement for all living organisms. So if you don't have a membrane, all living cells, yeah, if you don't have a plasma membrane, you don't have a cell, obviously. And its main function is to work as a selectively permeable barrier. Now let's spend a bit of time understanding these 3 words and what they mean, because it tells us a lot about the function of the plasma membrane. It is a barrier. A barrier, it's something that separates, yeah? And it separates the cytoplasm from the external environment. So it is something that is an obstacle, if you want, or it's a separator, yeah? But it's a barrier that is permeable. And by permeable, we mean that the barrier can be crossed, yeah? So there is the barrier, but molecules and substances can go from inside the cell to outside the cell and from outside to inside. So there is the ability to cross the barrier, okay? But this ability is not for everything and all substances, so it's selective permeable. So the permeability has some selectivity. Not everything can cross all the time. Some molecules need help to cross, some molecules can cross without help, but it is a selectively permeable barrier. So it is something that allows a physical separation between these two very different environments, the cytoplasm, the inside of the cell, and the outside of the cell. Because it is at the border with the external environment, it interacts with the external environment. The external environment is in contact with the plasma membrane, and its interaction with the external environment is possible because of proteins on the membrane that work as receptors, which can respond to stimuli that come from the environment. They can detect chemical, they can sense changes in the environment, and this detection can be interpreted by the cell in a way that makes the cell do an action in response. If it is sensing nutrients, for instance, present in the environment, the cell might decide to bring the nutrients inside. If it's sensing something which is dangerous for the cell might decide to move away and go somewhere else where there is no danger. So the receptors are there to sense the environment and respond to the environment. It works also as a transport system. As we said, it is a permeable barrier, so molecules need to enter the cell and need to leave the cell. And to do that, you need to have a system that allows for this movement. And this is done mostly through proteins, again, that are called transport proteins. And crucially, and this is a big difference compared to the eukaryotic cell. Sorry, I was looking for something, but I can't find it. Which is different from the eukaryotic cell. It is also the place where metabolic processes take place. Now, you know from general biology, I'm sure you've studied this, that some metabolic processes, important metabolic processes, need to generate energy by moving protons. Let's think about respiration. You remember respiration as it happens in the mitochondria of eukaryotic cells. Respiration is a process for the cell to obtain energy. It is a biochemical process which works by extraction of electrons, so oxidation of organic matter like glucose. Yeah, glucose becomes oxidized to carbon dioxide. The electrons are removed from the molecule of sugar. Yeah, and then these electrons, if you remember, are brought to an electron transport chain in the mitochondrion. And at the level of the electron transport chain, there is the movement of electrons that generates a gradient of protons The gradient of protons, which is the proton motive force, that is the key for making ATP, because it is the protons which activate ATP synthase and allow for the synthesis of ATP. This gradient of protons is possible because inside the mitochondria you have the cristae, you remember those membranes that are folded inside mitochondria, which allow for the formation of gradient. Now, if prokaryotic cells want to carry out respiration, they can do respiration. They actually do respiration in more than one way. So they have several different mechanisms for generating ATP using an electron transport chain. But they don't have mitochondria. And so the proton gradient, this pumping of protons to form the proton motive force that can activate ATP synthase, must happen on the plasma membrane, okay? So the plasma membrane is where any metabolic activity which requires the formation of a proton motive force takes place. So respiration happens on the plasma membrane. Photosynthesis, you remember how photosynthesis is also a process that requires a proton motive force. Photosynthesis also happens on the plasma membrane. Nitrogen fixation is something that only bacteria can do, which is taking nitrogen gas from the atmosphere and fix it into inorganic nitrates and nitrites that then can be changed into organic food matter, also happens on the plasma membrane. So you see, this is an added function. We have the selective permeability, we have the proteins, the interaction with the environment and all that. We have the transport system, but we also have metabolic processes taking place on the plasma membrane. What is the structure of the plasma membrane? It is very similar to the eukaryotic plasma membrane. It is a bilayer of lipids, phospholipids, bilayer with proteins scattered amongst the lipids. The lipids are amphipathic, so they have both hydrophilic and hydrophobic characteristics. You have the hydrophilic part of the molecule, which is the phosphate group, which is polar and interacts with water. Polar means that it has an electric charge. And then there are the hydrophobic non-polar tails, which are insoluble in water. Membrane proteins are peripheral proteins, like this one here, you can see here. This is proteins that are not very strongly attached to the membrane. They can be easily removed. Or you have integral proteins, which are proteins that cross the membrane. Because they cross the membrane, the part that is in the middle of the membrane must be also hydrophobic, yeah? And these ones are the proteins that are scattered in the membrane. There are sugars also, which can be sugars attached to the proteins themselves, in which case we say that this is a glycoprotein. Yeah, here you see these oligosaccharides attached to the protein. Or you can have sugars attached to the lipids directly, in which case this is called the glycolipid, okay? So again, we have you know, probably you're thinking, I know all these from studying the cell, even the eukaryotic cell, they're very similar. We have here is this phospholipids, the polar end. With the phosphate, you have the glycerol and the hydrophobic fatty acid chain. Now, one other point of difference when we compare the membrane of bacteria with the membrane of eukaryotic cells is the absence of sterol. You remember how in eukaryotic cell you have cholesterol in the membrane. The cholesterol is an important molecule in eukaryotic cell membranes because it works as a buffer for temperature. Meaning what? Meaning that cholesterol makes it harder to change the fluidity of the membrane, whether the temperature increases or decreases. Let's think about this for a second. Fat. We know the membrane is a lot of fat, yeah, a lot of DSM, yeah, and we know how fat changes its state according to temperature. So, for instance, if you take oil, yeah, olive oil, you know, zaytun, and you put it in the refrigerator, you put it in the fridge, after a while, you will find that the oil that was liquid becomes more like a solid. Yeah, it becomes denser. Butter, zibda, which is solid at room temperature, if you put it in the pan to make eggs, it becomes liquid. Yeah, so fats change their state, whether they are solid or liquid, according to temperature. Now, with the cell membrane, we're not going to look at liquid and solid. The membrane is a very fluid structure. It is in its consistency, it's similar to when you mix oil with lemon. You know, when you make salad and you mix oil with lemon, you get this kind of smooth fluid. It's not a liquid, but it's not a solid. Yeah, that's how the membrane is. And changes in temp, and this fluidity, this kind of almost liquid state of the membrane is what allows for movement. Remember, we said it's a permeable membrane, and it's permeable because it is fluid. If the temperature changes, if the temperature goes low, the membrane becomes less fluid. If the temperature goes up, it becomes more fluid. So if it goes down, there is less exchange. If it goes up, there is more exchange. Now, this change of fluidity happens with temperature, but cholesterol makes it harder to change the fluidity. So the temperature needs to change a lot because cholesterol is there in order to make the membrane change to the point that the permeability is affected, okay? And so cholesterol does this in the eukaryotic cell membrane, but bacteria don't have cholesterol. They have instead another molecule. They're called, these molecules are called hopanoids. Here is the picture. It's very similar if you look at the structure. You see the cholesterol has 4 rings, Here we have 5 rings, but is a similar structure to cholesterol and identical function. So it has the function also of working as a buffer for temperature. So having the hopanoid makes it harder to change the fluidity of the membrane if the temperature changes. Okay, do you have any questions? Okay, so let's move on. Okay, so this membrane is a barrier. It is a permeable barrier, so molecules move in and out of the membrane. Most of the molecules that cross the membrane are actually nutrients. Yeah, so it is food which needs to cross the membrane, enter the cell so that the cell can get energy and carry out its metabolic activity. So we are going to talk a little bit now about nutrients and how they move in the context of the plasma membrane. So here is the nutrients. What are nutrients? We can divide them into major groups. You have macro elements and micronutrients, macronutrients and micronutrients. Macro means big, micro means small. Now here, the use of macro is not so much having to do with the size of the nutrient itself, but with the quantity, yeah? So macronutrients or macro elements are the nutrients and the elements which the cell needs in large amounts, okay? So the macro is referring to the quantity. Examples of macronutrients are carbon, oxygen, hydrogen, nitrogen, sulfur, and phosphate. These ones can be all found in organic molecules like protein, lipids, carbohydrate, and nucleic acid. And then you have some minerals like potassium, calcium, magnesium, and iron. These ones are important for the working of some enzymes and some proteins. And these are the ones that belong to the macronutrients. Micronutrients are required in much smaller amount, trace amount. You have manganese, zinc, cobalt, copper, for instance. Often these ones are found in water. In the laboratory are essential for making up media to grow bacteria. And they are cofactors working for helping enzyme, yeah? Some unique substances might be required, like some amino acids, depending on different bacteria. They might be able to make a lot of amino acids, but not some of them. You remember the essential elements, essential components, which the cell cannot produce itself, yeah, so this is these are called growth factors, and the cell cannot make them, so that it must find it in the environment, otherwise the cell dies, yeah. And there are some amino acids which are needed for protein synthesis, some purines and pyrimidines. Again, it's not always the same for all species. Some can make one type and not the other. And vitamins also, again, not all of them, but some of them, they need to come from the environment. So how do cells collect these nutrients? What are the methods for uptake? Uptaking meaning bringing it inside. Yeah. So how does the cell do that? Well, first of all, they must be dissolved in water, yeah, and they need to cross the selectively permeable membrane. And the way that the crossing is done depends on the nature of the nutrient, whether it is big or small, and whether it is hydrophobic or hydrophilic. So small and hydrophobic molecules can cross the membrane easily without any problems, yeah? Large and hydrophilic molecules cannot. So how they cross depends on the nature of the molecule, hydrophilic, hydrophobic, large or small, but it depends also on the concentration. So if the molecule has high concentration outside of the cell and low concentration inside of the cell, it can move by diffusion. If the molecule has a high concentration inside the cell and low concentration outside, it needs to be taken in using energy, and so it is active transport, okay? Diffusion, which is the movement from high concentration to low concentration without needing to consume any energy, is of two types. You have passive diffusion and you have facilitated diffusion, okay? Passive diffusion is only possible for small and hydrophobic molecules. So small hydrophobic molecules, which are in high concentration outside the cell and low concentration inside the cell, can enter using passive diffusion. Molecules that also have high concentration outside and low concentration inside, but they're not hydrophobic and small, need to be helped. They need help to enter, and that is facilitated diffusion. It's still diffusion, it still doesn't need energy, it's still going from high to low concentration, but it requires protein to help, transporters to help the move. Active transport always requires proteins that bring the nutrient inside the cell against the concentration gradient, so going from low concentration to high concentration. You have primary and secondary active transport. And in bacteria, there is this other system, which is called group translocation, by which more than one molecules are transported at the same time, yeah, in a group. Yeah, that's why it's called group translocation. So passive diffusion, as I said, the molecule go from high concentration to low concentration between the cell interior and between the cell exterior and the interior. It goes inside the cell, high concentration outside, low concentration inside. Examples of molecules that can easily cross the membrane, as we said, small and hydrophobic. Water, H2O, can do that. Oxygen gas, O2, and carbon dioxide have the ability to cross the membrane without anybody helping, okay? They don't need proteins, they don't need transporter, they can cross the membrane on their own. For passive diffusion, the direction of movement is from high concentration to low concentration. Carbon dioxide, when it becomes too high inside the cell, goes out, oxygen goes in, and so on. Water has the ability to cross the membrane without the need of any transporter, but because when it comes to water, the cell needs usually to move quite a lot of water at the same time, there are actually specific channels in the membrane that are called aquaporins, which are dedicated for the movement of water, makes the movement of water even easier. Yeah. So that is diffusion. Facilitated diffusion is still diffusion. So it is still going from high concentration to low concentration. It's still not requiring any energy. Yeah. But the substance that is moved is either too big to cross, or it is hydrophilic, and so it cannot cross, yeah, because of the hydrophobic portion of the membrane. So it requires the help of proteins which facilitate, that's why it's called facilitated diffusion, makes it easier for the substance to cross the membrane. Because of this requirement of a transporter, facilitated diffusion depends on the concentration gradient. If the concentration gradient is high, so if there is a lot more outside than there is inside, the substance enters faster. Yeah. And it depends also on how many transporters are available. If all the transporters are being used, then the rate of transport decreases, or sometimes even reaches a plateau and doesn't and cannot go any farther, any faster, because of no transporters being available to move the nutrient. Okay, so the difference from passive diffusion is that it uses carrier molecules, which are called permeases. Permeases because they make the membrane permeable for those nutrients. The rate increases with the concentration gradient. If there is a larger difference, it moves faster. And it can be used effectively to transport glycerol, sugars, and amino acids. This graph here shows the difference between passive diffusion and facilitated diffusion. If you look at this graph, here on the y-axis, we have the rate of transport. So how fast is the molecule moving across the membrane, so rate of transport. And on the x-axis, you have the concentration gradient. So how big is the difference in concentration between the inside of the cell and the outside of the cell? You see, with passive diffusion, the rate of transport increases exponentially with the concentration gradient. You see, the concentration gradient increases, the transport is faster, yeah, and it goes up, 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 and it keeps on going up, yeah, it goes up. With the facilitated diffusion that requires a carrier, initially, the rate of transport increases exponentially as well. You see the concentration gradient increases and the rate of transport increases. It's slower compared to this because you need to load onto the transporter, you need to bring it in, you need to unload. So it has, it is a slower process, yeah? But it cannot keep on growing on a straight line. It slows down with the increasing concentration gradient until reaching a point, this is called a plateau, yeah, where the rate of transport stops increasing even if the concentration gradient increases. And this is because at this point, all the transporters are occupied. This is called saturation of the carriers. So when they're occupied, you can't bring any faster. Yeah, and so this is the plateau. Yeah. So both increase with the concentration gradient. However, the facilitated diffusion cannot continue to increase, and the rate of transport depends on the availability of free carriers. Yeah. This other side of the figure on the right shows you how a carrier protein can collect solute, yeah, and then bring it inside the cell, and then releasing the solute inside the cell. Are you okay with diffusion? Do you have any questions? Okay, so the next type of transport is active transport. Active transport also requires protein, like facilitated diffusion, but also requires energy, yeah, and it goes against the concentration gradient. So it doesn't go from high concentration to low concentration, it goes from low concentration to high concentration. It is an energy-dependent process. Energy can be supplied by ATP or by gradients of protons, the proton motive force. You remember what we mentioned before when we were talking about respiration and photosynthesis, pumping protons across the membrane creates a gradient of protons, and protons are positively charged, and so you change the electricity around the membrane, and this can supply energy for moving nutrients. So it is not only ATP that can be used, proton motive force can be used as well. And when it comes to bacteria, bacteria tend to use proton motive force more than ATP for energy, okay? So it moved molecules against the gradient, as I said. So it concentrates the molecule inside the cell. Uses carrier proteins as well, called permeases, and you have the same saturation effect that we saw with facilitated diffusion is also true for active transport, because with active transport as well, you might have the transporters being occupied. And so, you know, you have, if the concentration is high outside and higher inside, the transporters are not able to go any faster. Yeah. Now we're going to look at a couple, 2 or 3 examples of active transport in bacteria. We start by looking at one type of transport that is very common in bacteria, is actually not only present in bacteria, but is found also in archaea and in eukaryotic cell. This is called ABC transporters. Yeah, ABC stands for ATP binding cassette. Yeah, there is the protein has a portion, a domain of the protein which binds ATP. Yeah, so ABC transporters obviously use ATP and not proton motive force for energy. Yeah, they are made of two subunits, yeah. So two hydrophobic membrane spanning domains. Here is the transporter. You see this blue thing. There is a portion that goes across the membrane, and then in the cytoplasm you have the ATP binding part. The molecule that needs to be transported is attached to a solute binding protein. So the cell produces proteins that can collect the nutrient, and then the solute binding protein, which is loaded with the nutrients, attaches to the transporter, to the ABC transporter, and when that happens, ATP is consumed, energy is provided to open the gate, open the transporter, and allow for the molecule to come in, yeah? And so you have moved the molecule inside the cell. You might think, why don't we leave this open all the time? Well, this is a good question. Why don't we want to leave this open all the time? If we leave this open all the time, the molecule can go in without having to use energy. Can anybody answer these questions? Why do we want to use energy? And why don't we leave it just open all the time? Anyone has any idea? Because if the gate is always open, you cannot move against the concentration gradient. If the gate is open, the molecule will move according to concentration, and so it will be diffusion. Yeah, it will be going from high concentration to low concentration. But if you want to keep things in high concentration inside and low concentration outside, you need to close the door. And this closing and opening the door requires energy. So that was the ABC transporter. It's a primary active transporter. It's directly binding and bringing inside. Secondary active transporters use gradients of ions to co-transport substances. So here you have the uniporter, like the one that we saw before, the ABC transporter, uni, one thing coming in, but you can have symport and antiport as secondary transporters. If you have, for instance, here a proton, very often it is protons that are used, the ion is proton, but it can be also sodium. Na can be used as well as a type of ion. You can have them coming together in the cell, so going in the same direction. The ion goes in, and as the ion enters, it kind of drags the other substance with it, it pulls it with it, and this is the symporter. The antiporter, the two go in the opposite direction, so the ion goes out and the nutrient comes in, yeah? So symporter and antiporter are co-transporters, okay? Uniporter is just one thing moves, like we've seen with the ABC transport. It is ions, and very often these ions are protons or sodium ions. I told you before with this prokaryotic cells, we have another type of transport, which is called group translocation. This is not group, yeah, this is not group translocation. This is using the proton as a form of energy to open and close the transporter. With group translocation, you have large nutrients which are both that are moving together, yeah. It is an energy-dependent transport. During the transport, the molecules that are crossing are actually changed chemically. Most of the time, they are phosphorylated, so a phosphate group is attached to the molecule. A very good example of these systems is the phosphoenzyl pyruvate sugar phosphotransferase system, PTS. Yeah, it's been studied in great detail, and you can check. There is a YouTube link here if you want to see how this works. Yes, I did send the course slides on the ITC, which, you know, the ITC website for Birzeit University. If you can check whether you can access them there, I think it's easier for you to see them on the ITC site. But yes, we can make WhatsApp. I don't mind at all. I am going to write down my phone number here, 059787130. This is my phone number. I don't know how to make WhatsApp groups and things. You make it, add me, and I'll be happy to put the slides there as well. Okay. And also it would be make it easier also to communicate, and you know, if I have any difficulties, I can let you know, and we can organize ourselves better with the WhatsApp group. So here with the PTS, we have a more complex system. You see here there are several proteins that are involved. You don't need to worry about the details, but here you have two sugars, glucose and mannitol. They're both moved together at the same time, so it's a group translocation. They're brought inside, and because you see how these proteins here have phosphate groups attached to them, these phosphate groups are given to the sugars as they enter inside the cell, so that the sugars become activated. This is one way to start metabolism very fast. You remember how glucose in glycolysis needs to be phosphorylated for the process to start. If you bring glucose and you attach the first of the phosphates as it enters the cell, that sugar is already kind of ready almost to enter the metabolism and become, you know, enter the glycolytic pathway. Okay, so those are nutrients like sugars and large molecules. A specific mention should be done for iron. Iron is a very important metal which is required for respiration, for other metabolic activities, for some enzymes. But iron has a problem that when iron is oxidized, it is insoluble, Okay, so in the presence of oxygen, ferric iron becomes insoluble, it precipitates, and it's very difficult to collect this iron. And so microorganisms have a system to grab iron before it precipitates and help in the uptake, which is the siderophore. Siderophores, here is the iron is green. You see here the iron. The iron you see is attached to oxygen, here red oxygen, yeah. And by grabbing it like this, it stops it from precipitating, yeah. So siderophore complexes with ferric iron, and then this complex is transported inside the cell. And then even when it's inside the cell, it needs to be given to another something that holds it before it precipitates inside the cell. Okay, so this finishes the discussion of the membrane. We've said it is a semi-permeable barrier. It is composed of phospholipids and proteins with the mosaic model that you know already from the eukaryotic cell. We have hopanoids which take the same role as cholesterol. It is a semi-permeable barrier. It allows for movement of molecules, and we learned how these molecules can move. It is also the site of important metabolic activities like respiration and photosynthesis, for instance. And it is the place where receptors are located for cell sensing of the environment. So that was the first layer of the envelope around the cell. The second layer is the cell wall, okay? And as I said before, the cell wall is present in almost every single bacterial cell, unless the cells are parasitic, living inside another cell. If they're free organism, they must have a cell wall. And bacterial cell walls are there for some functions, okay? One of the function is the shape. It's rigid, it's a rigid structure, so it maintains the shape of the bacterium. It also stops the cell from breaking in case too much water enters the cell. Now you remember how cells might find themselves in environment that are either isotonic, hypotonic, or hypertonic to the cell. Isotonic is very rare, yeah. Cells, bacteria that live with us are mostly in isotonic environment, but otherwise the cells are either in hypotonic or hypertonic environment. Now, let me see if I can. I don't know how you do. I don't know. Anyways, hypotonic, it's an environment which is in which the concentration of solute is lower compared to the concentration of solute inside the cell. So if the concentration of solute is lower outside the cell, meaning outside the cell there is more water than there is inside the cell, so the water moves and goes inside the cell. When that happens, the cell grows in size because you have water, you accumulate water, the cell becomes bigger and bigger and bigger and bigger. and then the cell breaks. Yeah, this is called osmotic lysis. Yeah, this breaking of the cell due to an increase in size because too much water comes inside the cell. Now, if there is a cell wall outside that stops the cell from growing bigger and bigger and bigger, the osmotic lysis doesn't happen. Yeah, and so the cell wall protects against osmotic lysis in hypotonic environment. If the cell is in a hypertonic environment, where the concentration of solute is higher outside the cell than it is inside the cell, water goes out of the cell, there is plasmolysis, and the cell dies. Okay, so the cell wall doesn't help in hypertonic environment, but it can help in hypotonic environment. It is also another layer that needs to be crossed before getting to the cytoplasm, and so it protects also from toxic materials and the other elements that might damage the cell. In pathogenic bacteria, so bacteria that cause disease in humans, the cell wall might be one of the factors that cause us sickness. Okay, so it might contribute to pathogenicity, meaning that it might be something that damages our body or makes our body react in a way that makes us sick, gives us symptoms of sickness. Okay. What are bacterial cell wall made of? All bacteria cell wall contain peptidoglycan. It used to be called murine in the past. When I was at the university, they used to call it murine, but now murine is not used anymore. It is called peptidoglycan. The name in itself is already telling us what it's made of. It's made of glycan, sugar, and amino acid, peptide. Yeah, it is the rigid structure that is found outside the cell plasma membrane. So all bacteria have a cell wall. All bacteria have a cell wall that contains peptidoglycan. But how the cell wall is organized is different. You find the cell wall organized in two different ways. Either with a thick layer of peptidoglycan, so this substance is present and forming a big layer, and this is what we call a gram-positive cell wall. Or you have a thin layer of peptidoglycan, so a small layer of peptidoglycan with an outer membrane, and this is the gram-negative cell wall. Where does this name come from? Gram-positive, gram-negative. What does it mean? Yeah, it doesn't mean something is there or something is not there. Positive, negative is not in that sense. This is the name that has been given according to the way that the bacteria look if you color the bacteria, staining, coloring, sabra, yeah, if you give the bacteria color using a specific technique. This technique for coloring bacteria is called gram staining technique, and it follows very precise procedure. You take your bacteria, you fix them on a microscope slide, and then you color them using first a dye, which is called crystal violet. Then you wash the dye off, and then you put saffron in. You have to do specific steps. We're going to learn this when we do the laboratory part. And If the bacteria have a thick layer of peptidoglycan, they, at the end of the procedure, when you look under the microscope and you look at what you're seeing, you find the bacteria color purple, okay? And that's gram-positive. If the peptidoglycan is thin, the bacteria don't color purple, they color pink, and that's called gram-negative, okay? This difference in absorbing the dye of taking the coloring material is reflected in the architecture of the cell wall. So the way the cell wall is built determines whether the cell takes the purple color or the pink color. We're going to look at the structure in a minute. So what is peptidoglycan? I said it's peptido and glycan. Glycan meaning sugar. Peptido is the amino acid. Yeah. It is actually a disaccharide, so two monosaccharides joined together, N-acetylglucosamine and N-acetylmuramic acid, NAM and NAG. Yeah, these are the two sugars. Now, this disaccharide is repeated many, many times over. Yeah, disaccharide in a long line of disaccharide, and attached to the sugar, you have a short chain of amino acids. 1, 2, 3, 4, 5 amino acids. Yeah. L-alanine, D-glucamic acid, mesodiaminopimelic acid, and the last two are 2-D-alanine. Yeah. Now, I don't know what you are studying, but if you are studying pharmacy or pharmacology, these alanines are quite important in the working of some antibiotics and even in some antibiotic-resistant problems, okay? The amino acids are L-oriented or D-oriented. You might remember this from organic chemistry. Organic molecules can have right or left orientation. Yeah. So here you have the disaccharide chain and the amino acid, and then you have another chain with the other amino acid. This alanine here and the diapiminic acid there can be linked together by a peptide interbridge that is made of glycine. There is 5 glycines here that join these together. And so you have these sheets of sugar that are held together by amino acid chains, which are also cross-linked. And this makes very strong fibers, which make up the cell wall of bacteria. So this joining the sheet across using the glycine bridge is called cross-linking. Any questions? Okay, so let's look at the two different architectures. As I told you, gram-positive, gram-negative is a name that we give to two very specific ways of organizing the cell wall. With gram-positive cell walls, the peptidoglycan makes up most of the cell wall. Almost all of the cell wall is peptidoglycan. You see it here in this schematic representation. You see you have this thick layer of peptidoglycan. Here is the plasma membrane in red, and the peptidoglycan is this brown, thick wall. Yeah, very simple organization. Peptidoglycan might have teichoic acid, which is inserted into it. This teichoic acid has a lot of phosphate, so it is negatively charged, which helps the cell with keeping water on the cell. Yeah, teichoic acid also helps keeping everything together because it can hold the things together. Yeah, because it can cross the plasma membrane, and it might even bind to host cell. So here you have it, peptidoglycan with some teichoic acid. In some cases, you might find a layer of protein outside, but that is not very common. And it's found only in some of them. So there you go. Very simple architecture. It's just a big wall, basically, a big wall of peptidoglycan that is out there between the plasma membrane and the external environment. When you look, this is a drawing of the cell wall. You see here these are the sugars, yeah, the NAG and NAM, you see them? Here in black, small black lines, you have the amino acids that hold everything together. And then you have the teichoic acid, which might be attached to the peptidoglycan, or it might go all the way and link to the membrane and kind of holds the peptidoglycan onto the membrane. When that is the case, this teichoic acid is called lipoteichoic acid because it's attached to the lipids. Yeah. But you see, very, very simple structure. There is a small space between the plasma membrane and the peptidoglycan, and this is called the periplasmic space. Now, this periplasmic space is very small in gram-positive cell. It might contain enzymes for breaking down large molecules like starch or proteins. You know, starch is a very, very large polysaccharide. You can't, the cell cannot bring starch as it is inside the cell, but it can put enzymes in the periplasmic space that break down the starch to glucose, and then it brings the glucose inside. Yeah, these are called exoenzymes. These are the enzymes that now we use very often when we wash clothes. You know, when you put in your washing machine, you put the soap for the washing machine, very often it contains bacterial enzymes which break down these large organic molecules so that it's easier to take them away and move them from the material of the clothes. Yeah. So you see, it's a bit boring, the gram-positive cell wall. It's just a thick layer of peptidoglycan with a bit of teichoic acid. The gram-negative cell wall is a little bit more interesting because it doesn't only have peptidoglycan, it has an extra membrane. So here you have in red the plasma membrane, then you have the peptidoglycan. You see how thin the peptidoglycan here is? The brown, this dark brown line is very thin. But after the peptidoglycan, there is another membrane, which is called the outer membrane. So the cell wall is not just peptidoglycan, it's peptidoglycan together with an outer membrane. The periplasmic space, which is now between the plasma membrane and the outer membrane, is much larger compared to the periplasmic space of gram-positive cells, and so it is a space where the cell can make a lot of work. And also because it has another membrane, it's almost as if you had this kind of peripheral organelle around the cell. The outer membrane is a lipid bilayer, but is not only made of phospholipids. The inner layer, the one that is closer to the peptidoglycan, is phospholipids, but the upper layer is not phospholipids. The lipids are called lipopolysaccharides, LPS, and they're very different from phospholipids. So we're going to look at how that works as well. There is no teichoic acid in the gram-negative cell wall. So you see, when we look at the drawing of the gram-negative cell wall, we have a much more interesting set of parts. Yeah, here you have your plasma membrane with the proteins, blah, 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 the phospholipids. Then you have the peptidoglycan, very thin layer, maybe just 2 sheets of peptidoglycan are there. Yeah, so very thin. And here you have the outer membrane. And the outer membrane, you see, on the side that is closest to the peptidoglycan, you have phospholipid. But on the other side is lipopolysaccharide. Yeah, a lot of sugar. You see these lines that come up are actually sugar line. Yes, we're recording the lecture. I believe we're recording. We are recording, and I'll send you the link for the record. So you have the sugars, the polysaccharide. The membrane, it has proteins, and these proteins, some of these proteins make large holes. These are called porins. Yeah, they make holes which make the outer membrane less selective compared to the inner membrane. Yeah, and allows for material to enter the periplasmic space. Yeah, these are called porins. There are proteins, you see these proteins here called Brown's lipoprotein, which hold the outer membrane to the peptidoglycan, and then other proteins that hold everything together. As I said, the periplasmic space is very large. It can be up to 40% of the total cell volume. There are many enzymes, many activities can take place here in the periplasmic space. The peptidoglycan is very thin, yeah? 5 to 10% of the cell wall weight. Now, if we look at lipopolysaccharide, it is a fat and sugar, so it's lipids and sugar together. The lipid part is the hydrophobic part, the sugar is the hydrophilic part. The lipid is it's called lipid A, and then the sugar can be divided into 2 parts. The polysaccharide here, which is attached to the lipid, which is called core polysaccharide, and then a chain of sugars that is called the O side chain or O antigen. Yeah, the lipid is buried in the outer membrane, while the sugars stick out. And these sugars sticking out make gram-negative cell quite sticky, yeah, because sugars are sticky. And they can be recognized by the immune system, and that's why we call it O antigen. And we can use the properties of these oligosaccharides to distinguish between strains of bacteria that are of the same species. Like, for instance, E. coli, there might be E. coli that is more pathogenic than another E. coli. So an E. coli that can cause more disease than another E. coli. And you might be able to recognize it by looking at what the sugars in the lipopolysaccharide are. So this can be quite useful for us in diagnostic, from a diagnostic perspective. The sugars give negative charge, which also keeps the cell hydrated. Here is the O antigen, the core oligosaccharide, and the lipid A. Yeah? Host defense protection, the O antigen. The lipid part is actually a toxin for us. If we get infected with gram-negative bacteria, the lipid A is an endotoxin that might be responsible for causing some symptoms like fever, headache, dizziness, vomiting. Yeah, this might be caused by release of lipid A in our body if we get infected with a gram-negative bacterium. As I said, the permeability of the membrane is also an important factor. Now, we already discussed how it can be hard to cross the plasma membrane because it's a selective barrier. Imagine having to cross two membranes. It makes it even harder. And this could cause problems when the cell needs to get nutrients. And so in order to avoid too many constraints for nutrients to get to the cell, the outer membrane is more permeable compared to the inner membrane, compared to the plasma membrane. And this increased permeability is possible because of the presence of proteins that make big holes in the membrane so that molecules and proteins can, and nutrients can enter in an easier way. Yeah, these are called porine proteins and transporter proteins. So they, as you see here, they form channels that allow for molecules to get into. They don't get into the cell, they get to the periplasmic space, and there then they have to cross the plasma membrane to get to the site. Okay, we said this before. We talked about cell wall and osmotic protection. I said that the cell wall protects the cell in hypotonic environment. A hypotonic environment are environment in which the solute concentration outside the cell is less compared to the inside of the cell. So water comes inside, the cell gets bigger and it breaks. Yeah, and the cell wall protects from this lysis. Hypertonic environments are environments in which the concentration of solute outside the cell is more compared to the inside of the cell. So water goes out of the cell. This causes plasmolysis, and the cell might die. The cell wall does not protect in a hypertonic environment. It only protects in isotonic environment. And you can see this in an experimental way. If you treat a bacterial cell with a enzyme, lysozyme, that breaks down peptidoglycan, the cell doesn't have a cell wall anymore, doesn't have peptidoglycan anymore. If you treat cells either with lysozyme or with penicillin, which is another, which is an antibiotic that stops the building of the cell wall, you have a cell without a cell wall. This is the cell with the cell wall. Without the cell wall is called the protoplast. Yeah, see, it changed the shape because there is no rigid cell wall around it anymore. Now, if these cells are kept in isotonic environment, they're okay. They don't. They can stay. Yeah, and you can have protoplasts, and they, as long as you supply them with nutrients and temperature and things, they're okay. But if you transfer this protoplast to a hypotonic medium, water will enter, you will have swelling, and then you will have breaking of the cell. So cells that don't have a cell wall might survive in isotonic environments. And do you remember we mentioned at the beginning of this lecture mycoplasma? Mycoplasma is a parasitic bacterium that lives inside the cytoplasm of eukaryotic cells. A cytoplasm and the cytoplasm, they're isotonic, so it doesn't need to have a cell wall. So mycoplasma never produces a cell wall. Actually, mycoplasma is interesting because mycoplasma, because it is always in contact with the eukaryotic cell, actually is one of the very few bacteria that has cholesterol in its plasma membrane. Probably it learned from the eukaryotic cell to use cholesterol, yeah? And the plasma membrane is a bit more resistant to osmotic pressure, but it lives in a isotonic environment. Okay, so that is the cell wall. Is it okay? Do you have any questions? Right, so now we move to looking at other structures that might be found outside of the cell wall. Just give me a second, I'm going to get a bit of water, yeah? Right, so after the cell wall, we might have other structures. Again, it is not a requirement. Some cells have it, others don't. It depends on the species. It depends on the structure of the cell. What do we have? We have capsules and slime layers. These layers outside, which help with the cells becoming sticky and attaching to surfaces, called glycocalyx. Glyco, again, sugar, yeah? Capsule and slime layers are made of sugars. But there is also S layers. S layers are not made of sugars, they're made of proteins, yeah? Now, when cells have these layers of sugars surrounding the cell wall. Sugars, we know, is sticky, and it can help cells coming together and living next to each other, forming a community of cells, which is an organization that we find quite often with free living bacteria, which is called biofilm. We're going to talk about biofilms later in more detail. Just here they are mentioned because of the sugars, which are necessary for holding cells together. So, capsules are saccharides, they're sugars, polysaccharides. Capsules are very well organized. They're difficult to be taken out of the cell. And you can observe them under the light microscope using negative staining. You see here, do you see these white circles around the cell? This is the capsule. Yeah. It protects the cell. Having sugars around it makes it less visible, for instance, to the immune system. Again, if we get infected with a bacterium, if the bacterium has the capsule covering the surface of the bacterium, our immune system finds it harder to find these cells, so it makes it resistant to phagocytosis. It's very sticky, it attracts water, and so keeps the cell hydrated, protects from desiccation. It's another layer to cross, and so some viruses, phages, for instance, cannot cross it. Detergents find it harder to cross, soaps, and even antibiotics might not reach the cell, so it's a protective layer. And it is a source of nutrients. If the cell is desperate and there is no food, it can consume the capsule for energy. The slime layer is very similar to capsule, same functionality, but is not as well organized as the capsule, and so it's easier to remove from the cell. S layers, as I said, are not sugars, they're protein. It can be glycoprotein, so it could be proteins associated with sugars. These are proteins which can self-assemble. What do we mean by self-assemble? These proteins work a little bit like crystals. I don't know, I'm sure you've taken some chemistry courses where you have talked about crystals and how salts in water, after they reach saturation point, they kind of collapse onto each other to form crystals that are organized structures. And the S layers work roughly in the same way. They are protein or glycoprotein, which organize that when they are together, they kind of come together forming regular structures. Yeah. If this happens in the gram-positive bacteria, is right after the peptidoglycan. If it is in gram-negative bacteria, is attached to the outer membrane. They have a function that is similar to the one of capsule and slime layer, so they protect from ion and pH fluctuation, osmotic stress, enzyme and predation. So they are an extra layer that protects the cell, maintains shape and rigidity, attaches to surfaces, again, keeping cells sticky, and protects from host defense. Now, some of these proteins are now used in biotechnology, nanobiotechnology, to build structures with a specific shape by putting these under specific conditions so that you can control the shape and produce tubes, protein tubes and other things that can be used, for instance, in medicine, that can be used for surgery, that can be used for making materials that are very flexible. Yeah. And with this, we finished the layers which are found outside of a cell, moving from the plasma membrane. You have the cell wall, and then you have these other potential places, the capsule, the slime layer, and the S layer. Now we go back to inside of the cell and we look at what is found in the cytoplasm. As I said, much simpler, no organelles, no internal membranes, very little in the cytoplasm of bacteria. You have cytoskeleton. This is wrong. Intracytoplasmic membrane gives the wrong idea. I will explain what they are in a minute. You have inclusions, ribosomes, the nucleoid, and you might have extra DNA in the form of plasmid. So we start with the cytoskeleton. The cytoskeleton is proteins which help giving the cell its shape and moving things inside the cytoplasm. They help in the cell division, making sure that the cell divides correctly, so in the right position where you have the breaking and the division of the cell. It helps also with the separation of the DNA during cell division. So functions which are very similar to the function of the cytoskeleton of eukaryotic cells, so much that a lot of the proteins that belong to the cytoskeleton of bacteria are homologs to actin filament, microtubules, and intermediate filaments. What do we mean by homolog? They're very similar. They're not identical, but they're very similar to the proteins of the cytoskeleton of eukaryotic cells. They work in cell division, as I said. They work in fixing protein in a particular place, moving things around in the cytoplasm, and determining the cell shape. Again, we could talk about the cytoskeleton of bacteria for a week, but I'm just giving you a few examples of cytoskeletal molecules that are found in bacteria. These are probably the best known ones are the FTS protein. FTS stands for filamentous temperature sensitive, FTS. There is more than one FTS protein. FTSZ is one that is found in many bacteria. And here it's in this picture is this green color here is the FTS protein. This is during cell division. It helps the cell with finding where to break to make two cells. Yeah. There is MREB, which is found in many rods, which is important for shape and keeping the peptidoglycan synthesis correct. CRES is not as common as these other two. This is found mostly in Vibrio to maintain this curved shape. Yeah. So these are a couple of examples. Yeah. Now, These that are called here intracytoplasmic membranes, they're not membranes that are found inside the cytoplasm. They are actually folds of the plasma membrane. So, you know, the plasma membrane, you can imagine it as a circle going around the cytoplasm. If you remember what we said earlier, we said that the plasma membrane is also the place where you can have metabolic activities like respiration and photosynthesis, which require pumping of protons and forming gradients. Now, if the membrane in these bacteria, only these bacteria, if the membrane was limited to just the circumference of the cells, covering the cell surface, the cell will not have enough space and enough membrane to have efficient metabolic processes. So the membrane is actually folding many times over, like what you studied in the mitochondria and in the chloroplast. You remember how mitochondria and chloroplast have folded membrane to increase the surface for efficient respiratory and photosynthetic process. The same is here. It looks like these membranes are inside the cytoplasm, but they're not inside the cytoplasm. This is the plasma membrane folding many times over to form a large surface area for the metabolic activity. So basically here you have the cytoplasm going around this membrane. Yeah, we find this organization of the infolded membrane in bacteria that do photosynthesis, and there are many bacteria that do photosynthesis. The cyanobacteria produce an enormous amount of oxygen because they're very efficient photosynthetic bacteria, and bacteria that do a lot of respiration. So these bacteria will have this folding, which increases the surface of the plasma membrane so that you can have the proton motive force, the ATP synthase, and the efficient metabolic process. One of the structures that also might be found in the cytoplasm is inclusion bodies. They're granules, crystals, or globules of organic or inorganic material. They're storage. Yeah, the cells bring in the food, bring in the minerals, bring in whatever they need, and they store it in these kind of crystals or masses of organic or inorganic material. Sometimes they surround them with something that separates them. It's not a double membrane, it's not even a proper membrane. Sometimes it's just proteins which kind of are put around these things in order to separate them from the rest of the site. The storage inclusion, we find storage of nutrients, storage of metabolic end products, like for instance, amino acid, energy, building blocks. They can have glycogen storage, sugar, yeah. For carbon storage, often carbon is stored, glycogen is for storing glucose. For storing carbon, they can use polybeta hydroxybutyrate, which is a way to store carbon. You see here you see this surrounded by protein to make this polybeta hydroxybutyrate contained, surrounded with protein. You can have phosphate polyphosphate. This is sulfur globules. You can have amino acid, cyanophycin granules, yeah. Or sometimes you find that without having a membrane, so not an organelle, but sometimes you find that some components of the cell are kept together all in the same area. Yeah, these are called micro compartments. For instance, for photosynthetic bacteria, the enzyme Rubisco, which is the carbon fixing enzyme for producing sugar using the energy from the sun, is concentrated in these areas of the cytoplasm. You can see here in this electron micrograph picture different consistencies of the cytoplasm, because this is where all these enzymes are grouped together. Yeah, these are called carboxysomes in carbon dioxide fixing bacteria that contain the enzyme Rubisco for carbon dioxide fixation. Bacteria that live in water, aquatic bacteria, and photosynthetic bacteria might have inside these vacuoles protein balls, as if it were, which might be filled with air or emptied of air. When they are filled with air, gas vesicles, when the bacteria puts a lot of air in this, makes the bacteria less dense and so able to go up in the water. When they are empty of air, they go down. And another interesting type of inclusion body is the magnetosomes. Magnetosomes have magnetite particles which help bacteria finding the Earth magnetic field. You know how the Earth has the north and the south magnetic field. And you have this magnetosome. You see them, they come ordered in a line. This lined way is through a cytoskeletal protein, which is called MAMK, yeah, which helps forming this magnetosome chain. What do bacteria do with this is not clear yet. Why do bacteria need to know the orientation of the Earth magnetic field is something that is not clear, might be related with finding food, but is not very clear what is the role of the magnetosome. Ribosomes are essential for living cells. They are necessary for the synthesis of proteins. In bacteria and in archaea, the ribosomes are a little bit smaller and less dense compared to the eukaryotic ribosomes. So they are given the measure of 70S. Eukaryotic ribosomes are 80S. The whole ribosome is 70S, but ribosomes are made of two subunits, a large subunit and a small subunit. The small subunit is 30S, the large subunit is 50S. Ribosomes are made of protein and ribosomal RNA. The ribosomal RNA that is found in the small subunit is a 16S ribosomal RNA. In the large subunit, you have the 23 and 5S. Now, I think this link for this YouTube video that shows the difference between 70S and 80S ribosome can be quite interesting. This S is for the Svedberg unit, and it basically measures density. Yeah, so 80S ribosomes are denser than 70S ribosome. It might be a bit confusing that the 70S ribosome has 50 and 30 S subunit. If you put 30 and 50 together, you get 80, but that's not how it works. It's 30 on its own, 50 on its own, and 70 when they are together. For the eukaryotic ribosome, the subunits are 40S instead of 30S, 60S instead of 50S, and the whole ribosome together is 80S. Ribosomes are for the protein synthesis. Then you have the chromosome, the nucleoid, not a membrane. There is no membrane around it, so this is prokaryotic cells don't have a proper nucleus. It is the place where you find the chromosome with its associated protein. The chromosome is present in one chromosome, so there is no pair of chromosomes. It's a single chromosome, so you don't have diploid, haploid. Your prokaryotic cells are always haploid. Yeah, there is no two copies of the same chromosome, just one copy. Yeah, one closed circular double-stranded DNA molecule. The DNA needs to be coiled and supercoiled in order to reduce the size of this large molecule to a volume which can be contained inside the protein. And the coiling and supercoiling is done using proteins that allow for this condensation. I'm going to see if I can find something here which I may use to explain this idea. Okay, let me see what I have here. I have this. Okay, so if this is the DNA, you see here I have this, it's very long, it's a very long piece of cable that I have here. Now, if I want to fit this in a very small space, in order to do that, I need to condense it. I need to make it smaller. And if this was a protein, yeah, one of these proteins, I can take the DNA and I can wrap it around the protein. You see, when I wrap it around the protein, now the size has decreased, yeah? And this is the job of these histone-like proteins to kind of condense and make the volume that is taken by this very large molecule, even though bacterial chromosomes are much smaller compared to our chromosome, they're still, 4 million base pairs, so they're quite big. Here in this figure here, you see here at the bottom, this is a bacterium, and this yellow line that you see outside is the chromosome when it's not condensed. You see, it's a much bigger the space than the space of the bacterium. So you need to condense it in order to fix it. The chromosome contains all the genes which are necessary for that species of bacteria to carry out its all metabolic activities. All the essential genes are on the chromosome, and the metabolic activities, or the shape, whether there is a capsule or not, whether there is a flagella or not, whether it is a gram-positive or a gram-negative, it's all on the instructions on the chromosome. There is extra DNA that might be present in bacteria. These are much smaller circular DNA molecules. It's outside of the chromosome. It's called extrachromosomal RNA, DNA, which are plasmids, yeah? And plasmids are independent from the chromosome. They don't carry any essential instruction. So if the cell doesn't have plasmid, the cell can keep on growing, dividing, and surviving. If they have a plasmid, if the plasmid is there, the plasmid might bring some extra activities, some extra abilities, which always make the cell better, okay? So plasmids are good for bacteria, but they're not essential, yeah? Sometimes plasmids can enter the chromosome, they can integrate into the chromosome. In that case, they're called episomes, okay? Now, what these plasmids are and what they do is not here. Oh, okay. You have plasmids which might carry the ability to resist antibiotic, or they might give extra metabolic activities, or they might allow to exchange other plasmids. So this is all abilities which are an advantage to the cell, but they're not essential for survival. So that's it. We finished the cytoplasm as well. So as you can see, not much there. Beyond the cytoplasm, beyond the cell wall, beyond the capsule and the limelight and so on, you might have external surfaces, external structure. Basically, pili and fimbriae and flagella are the possible structure. These structures are there if the instructions to make them are found on the chromosome. Some cells have it, some other cells don't. Fimbriae and pili. Fimbriae are short. You see here in this picture, this short sticking out lines, short, thin. They look like hair. They're made of protein. Their main function for fimbriae is attachment. So they help cells to attach to surfaces. Yeah. Sometimes they can help with movement, and they can also be used to pick up DNA. Bacteria are very good at collecting DNA from the environment, and they can use fimbriae to do that. Pili is these darker lines here. They're longer, you see, they're much longer compared to the fimbriae. They're thicker. There is less of them. Fimbriae can be up to 1,000 in a cell, pili 1 to 10. They can be made only, the sex pilus particularly can be made only if there is a conjugation plasmid inside the cell that gives the instruction to make it. And then this can be used as a bridge with another cell, like here, you see here, it can be used for a bridge to this other cell to transport, well, actually, no, this is wrong. It should go into the other cell directly to transport, like here. Here, you see how it makes this bridge to transport plasmids. And so they can be used to exchange plasmids between bacteria. Flagella are much longer. They are also made of protein. The protein that makes flagella in bacteria is called flagellin. Yeah. Flagella are for movement mainly. They can be used to attach to surfaces, but mostly is movement. Yeah. And They can be, you can find one flagellum, monotrichous is one flagellum. You can have the flagella, two flagella at different ends of the bacteria, polar flagella. They can be amphitrichous, so flagella are all at one end and there is nothing at the other end. You can have lophotrichous, or you can have peritrichous, where it's spread all around this. They are complex structures made of flagellin. The organization of the flagellum is different in gram-negative and gram-positive cell. With the gram-negative, the flagellum needs to cross two membranes, yeah, the plasma membrane and the outer membrane, while in the gram-positive, that is not a problem that the flagellum has. So you see how in the gram-negative, the structure that holds the flagellum, that is called the basal body, is more spread and larger in size compared to the one of the gram-positive cell. And there are all these rings, the L ring, the P ring, and the C ring, and the MS ring that are making the basal body of gram-negative flagella. With the gram-positive, you have a much simpler structure. Yeah. Then there is a hook, it's like a curved bit, and then you have the actual flagellar filament coming out of it. Now, these rings here, which are part of the basal body, they work like rotating rings that make the flagellum move. Yeah. Here you have the example of how the flagellum is made in gram-negative cells. You see, you have the outer membrane, the peptidoglycan, and the inner membrane. You have the ribosome produces the flagellin, and then the flagellin enter the basal the basal body and is pushed through here, like almost as a syringe that puts the flagellin outside and increases the size of the flagellum. Flagella are used for movement, yeah, and different rotations cause different actions. A anti-clockwise rotation, so the flagella going opposite the direction of a clock, yeah, makes cells go forward. So the flagellum works like a propeller, yeah, and makes it go forward. A clockwise rotation, so going in the same direction as a clock, makes the cell stop and stumble and tumble and change direction. Yeah. Why do bacteria want to move? Most of the time, bacteria want to move because they're looking for food, and so they are what pushes movement is what we call chemotaxis. Yeah, so it is movement based on chemical. Taxis is movement. Yeah, chemotaxis, and flagella can go towards chemical attractant or away from harmful substances. But that's not the only reason for movement. Bacteria might move because of temperature, light, oxygen, osmotic pressure, gravity, all sorts of reasons for movement. And as I said, the flagellum rotates like a propeller counterclockwise, so against the clock, makes the cell go forward while clockwise stops and makes the cell stop. And when the cell stops, it can change direction. Yeah, it can move in a different direction. Now, to move, you need energy. Yeah, and also flagellar movement requires energy. This is another bacterial activity that doesn't use ATP for energy, but it uses gradient of protons, proton motive force. Yeah, as I told you before, it is the rotation of the rings, these protein rings, which allow for the movement. You have a rotor and a stator. The C ring, you see this one, the C ring is the rotor, and the mot A and mot B are the stator. Here is mot A and mot B. Yeah. What happens? You have the cell forming a proton motive force. So protons are pumped from the membrane into the periplasmic space. You remember we said the space between the membrane and the peptidoglycan is the periplasmic space. And then the proton comes back inside the cytoplasm, going through this space between mot A and the MS ring. Yeah, it goes here, it goes through. And when the proton comes in, it pushes the ring and it makes the ring turn. Yeah. So the proton motive force is what allows for the movement and the rotation of the flagellum. As I said, chemotaxis is the movement towards chemicals, yeah, or away from a chemical. It usually works on gradients, and so if there is more concentration of the chemical, here you see it's darker green, the cell kind of goes in that direction. If there is no gradient, the cell keeps on trying to change direction until it finds a direction towards which it can go. So changing concentration of chemical attractants and chemical repellent bind chemoreceptors on the surface of the cell and kind of direct the cell in a specific way, which is towards the food source. And the last thing which we need to discuss today is the endospore, yeah, the bacterial endospore. Bacteria, not all of them, again, only few bacteria that have the ability, because in their genome, they have the gene that give the instructions to do so, can, if necessary, transform from a cell that is living and carrying out metabolism, growing, dividing, eating, and doing all those things into a non-metabolically active, sleeping, dormant structure, which is a survival structure, allows the bacteria to stop all the metabolism, all metabolic activities, covered itself in layers, protective layers, and stay there waiting for the dangerous conditions to pass so that it can come out again and start growing again. What are the conditions that might push a bacterial cell to transform into an endospore? It could be heat, change in temperature. so much heat that it might denature the protein and kill the cell, radiations, chemicals, lack of water, lack of nutrients very often, yeah. So when that is the case, the cell kind of says, okay, this is no longer a place where I can survive, I can grow, I can divide. I turn into an endospore. I wait for good time to come back. When that happens, I can go back to metabolism, to being a vegetative cell, is called a cell that is carrying out all cellular activities. It does this process by using calcium complexed with dipicolinic acid to remove water from the cell without destroying the DNA and the protein. Again, also DNA binding protein, which kind of hold and keep the DNA from being degraded. Loss of water, so that metabolism is zero, yeah, and a layer of coats, a layer of tabakat on top, which protect from the environment. So if you look at the endospore structure, you have the core, here is the inner membrane, and then there is the cell wall, and then there is cortex, outer membrane, coat, and exosporium. All these are many layers which are very thick, very difficult to penetrate and protect the cell. The core has very little water, it's like a gel. It contains the DNA and the ribosomes. It needs to keep ribosomes, the cell, for when it starts growing again, it needs to make proteins, yeah. Becoming an endospore is not easy. It's a long process. It takes up to 8 hours. Normally it starts when there are no nutrients, and it's a multi-stage process. The cell divides, and then it starts, you see this invagination of the membrane, forming 2 separate compartments, yeah. This one is going to die, the cell here, the DNA here is broken, yeah. But this one starts adding layers and layers and layers, and you start having this loss of water, condensation, gel-like consistency of the cytoplasm, until you have completed all the layer and you have a free endospore. This endospore can stay for years and years and years. They have found in Egypt endospores in mummies, which were there for 5,000 years, and when they were put into nutrient broth and things, they started growing again. So they're very, very resistant forms of the cell. The opposite, going back to being a cell that grows, divides, and carries out metabolism, which is called germination, is much faster. It takes minutes. As soon as the endospore senses the changes in the environment using receptors and water and all these things, the germination process takes only a few minutes. So making the endospore takes hours. Going back to a vegetative cell takes much less, and the cell can restart growth, division, and all that. And this is the end of the chapter. So I think it's also the end of our lecture for today. I will send the other chapter on the ITC for tomorrow. Tomorrow we have our lecture from 6 in the evening until 8. I will send you a message on ITC as well for that. Okay. Thank you, Nada, for staying all this time, and I will see you again tomorrow, inshallah. I will send a recording of this lecture later on. Bye.